In this chapter, we will provide a brief introduction of electroencephalography methods and the event-related potential. Electroencephalography, or EEG, is a technique for measuring electrical signals in the brain via recording electrodes placed on the scalp. Before we go over the general procedures of EEG, it's important to know what kind of information EEG is designed to measure. Individual neurons undergo changes in membrane potentials in order to pass chemical signals between one another by way of neurotransmitters. When neurotransmitters are released from the presynaptic cell and bind to receptors on the postsynaptic cell, this causes ion channels to open or close. The resulting change in voltage across the membrane of the postsynaptic cell is called a postsynaptic potential. Under conditions of mass synchronization across a population of neurons, it is possible to record the summation of postsynaptic potentials at the scalp using EEG electrodes. During an EEG session, recording electrodes are first inserted into what is called a cap or net. Electrodes are arranged in a specific way that is constant across participants and studies, and each electrode is labeled by its position on the scalp. For example, F stands for frontal, C stands for central, P stands for parietal, and O stands for occipital. The electrodes are connected to an amplifier so that the signal can be observed and interpreted by the technician. The technician prepares to apply saline-based electrolyte gel to the participant's scalp in order to facilitate the connection to each electrode. After placing the cap on the participant's head, the technician applies a small amount of gel to the area between the scalp and each electrode. The changing colors of the electrodes indicates the impedance, or resistance to current flow, which must be reduced below a pre-specified value in order for the signal to be of sufficient quality. After the technician verifies that all electrodes are receiving signal, a computer that is connected to the amplifier records continuous electrical output while the participant completes a task. Electrical activity is measured by EEG in the form of waves that are characterized by rhythmic aspects such as frequency and amplitude. Frequency is measured in hertz and indicates the number of peaks per unit of time. Amplitude is measured in microvolts and indicates the strength of the electrical signal at a particular time point. Changes in amplitude are often of interest when studying differences between states, such as task engagement relative to resting state, or when studying how the brain responds to experimental events such as stimulus onset, response selection, or feedback. As we will discuss next, amplitude changes that occur in response to an event are called event-related potentials, or ERPs. As an example, consider the classic oddball experiment in which a participant passively observes a sequence of trials consisting of 80% X's and 20% O's. At the end of the experiment, we can take the continuous time series of voltages recorded from each electrode and align it to the progression of X and O stimuli that were presented to the participant. To conduct an ERP analysis, we first segment the voltage data that was recorded while each stimulus was on the screen and time-lock it to the time of stimulus onset. We then average the time-locked waveforms across trials within each condition. By comparing the ERPs between conditions, we can observe the typical oddball effect of a higher amplitude peak for rare compared to common stimuli between 350 and 450 milliseconds post-stimulus onset. By conducting the same analysis at each electrode and computing the average difference within the time window of interest, we can visualize the effect as a topographic plot with a peak at electrode PZ. With the ERP technique, we can provide evidence that there are neural processing differences associated with common and rare items that are independent of the physical characteristics of the stimuli themselves. Limitations of EEG techniques are primarily related to their poor spatial precision. Because electrical signals need to pass through the skull and be considered an aggregate, it is difficult to identify the precise brain region from which a signal originated. Additionally, EEG techniques are limited in their ability to record activity from deeper structures beyond the cerebral cortex. Nevertheless, there are many advantages to using EEG techniques in neuroscience experiments, including its low cost for equipment and materials, its temporal precision on the order of milliseconds, and the fact that it is safe and non-invasive for use on participants of all ages and most clinical groups.